Good afternoon, everyone. I'm a professor of microbiology and environmental toxicology, and I'm, excuse me, and I'm also the associate faculty director of the UCSC Chemical Screening Center, and I lead an interdisciplinary research consortium that seeks to identify natural products that could be the next generation of antimicrobial drugs. So in the first part of the 20th century, a large proportion of the US population died of infectious diseases. But then in the 1940s, we started treating bacterial infections with antibiotics, and this contributed to a significant increase in life expectancy. So now in today's population, our deaths are mostly from non-infectious causes, and we expect that when we get sick, we can take antibiotics to treat our bacterial infections, right? And this is largely thanks to natural products. So natural products are chemicals that are produced by living organisms that serve a biological purpose. And more than half of all the antibiotics that we currently use in the clinic are derived from natural products that come from microbes. So for example, the penicillium fungus, which lives in the soil, produces and secretes the antibiotic penicillin. And penicillin itself doesn't harm the fungus, but it targets and kills bacteria in the surrounding environment. And this then allows the fungus to have access to more nutrients, and then this promotes the fungus's survival. So we've learned from this microbial competition, and we now use antibiotics that are originally derived from microbes in order to increase our own survival. So the discovery of penicillin really kicked off this what's called the golden age of antibiotic discovery. So the majority or almost all of the antibiotic classes that we use in the clinic today were discovered up until the 1990s. But antibiotic drug discovery is not very lucrative financially. And the last new class of antibiotic that's in the clinic was discovered in 1987. So we've had this antibiotic discovery void, and so no new classes of antibiotics are entering the clinic. And in addition, we haven't been training scientists to actually do antimicrobial drug discovery. So this is a looming problem for all of us, and this is because bacteria can become resistant to antibiotics through mutations or through uptake of antibiotic resistance genes. And this means that a growing number of infections can't be treated with antibiotics. So in 2019, there were more than 1 million deaths worldwide that were attributable to antimicrobial resistant infections. And it's predicted that we don't introduce new antibiotics into the clinic, by 2050, there'll be more deaths per year from antimicrobial resistant infections than from cancer and diabetes combined. So clearly, we need new solutions. So classic antibiotics target both beneficial and pathogenic bacteria, and this actually has unintended negative consequences. But what if we could identify new classes of antimicrobial molecules that only target pathogenic bacteria? So pathogenic bacteria often have unique surface structures that are unique to pathogenic bacteria, not beneficial bacteria. And these are often important for causing infection. So for example, I'm showing you here this bacterium Pseudomonas, which causes pneumonia and other types of infections. And you can see that it's got this long corkscrew type structure that helps it move, and it has these hair-like fibers, and those help it attach to surfaces. So those are examples of surface structures. So what if we could actually find antimicrobials that block surface structures specific to pathogens? We should be able to then treat infection without actually perturbing beneficial bacteria. So a surface structure that's used by dozens of pathogens to cause disease is called a molecular syringe. And in this image, I'm showing a cross-section of a bacterial cell where the two membranes are in green, and these molecular syringes are shown in blue. So pathogens like Pseudomonas and Salmonella use these molecular syringes to inject toxins directly into human cells, and this allows the bacteria to escape immune detection and immune defenses. So the goal of our consortium is to identify antimicrobials that block this molecular syringe. And we work under the hypothesis that nature has already evolved such chemicals. We just need to find them. So the strength of our team is that we have expertise both in chemistry and in biology. So our consortium's natural product chemists mine aquatic and terrestrial environments and look for organisms that are rich in natural products. They then crack open these organisms and they take the natural products that they make and they catalog them into these tiny plastic chambers, which then can be frozen down and used as a library. The consortium's microbiologists develop experimental techniques that allow us to test whether a natural product can actually inhibit the molecular syringe. And also the robotics in the UCSC Chemical Screening Center allow us to then do thousands of these kinds of experiments every week. So far, our consortium has identified several classes of chemicals that block molecular syringes, and we're busy validating the activity of those and developing them further. And importantly, we've also been able to train dozens of students as well as postdocs who now are going on to scientific careers armed with expertise in antimicrobial drug discovery. But it's still early days, and the path to the clinic can be rocky. And one of the pressing things that we always come up against is funding. So we've been lucky enough to have funding from the National Institutes of Health. However, they have limited funding streams for antimicrobial drug discovery because it's a risky venture. But I think at this point we can all agree that given the magnitude of the antimicrobial resistance problem, we can't really afford to ignore these possibilities of new therapeutics. We're also now doing experiments to test the efficacy of our compounds in preclinical models. And we do need to determine whether indeed these compounds inhibit the pathogens and leave the beneficial bacteria alone. And lastly, down the road, we'll have to pair these targeted precision drugs with actually precise molecular diagnostic tools to make sure that the infections we're treating actually involve a molecular syringe and we can treat that. So I want to acknowledge the critical contributions of the other people who have done the work with us. Phil Cruz, Scott Lokey, Roger Linnington, Jared Shaw, and John MacMillan. Their labs have been really instrumental in helping us do this really collaborative work. And also there's dozens of students and postdocs, not all of which are shown here, and I want to thank you for your attention as well.